Okay, all right. Okay, okay. See, botany, madam, zoology, sir, they will also teach the complete syllabus. They'll do botany separately, they will do zoology separately, whereas I will do complete biology syllabus. Okay, as a result, you can revise the entire biology 2 times. Okay, so the ultimate goal of teaching is, we'll keep the target of a realistic target. Okay, 1 second. Yeah, so we keep the realistic target of 340 out of 360. Okay, which can be doable easily. Okay, but in order to get that extra 20 marks, we have to answer those questions that cannot be answered by anyone. For that, only we have to read extreme depth of the concepts. The more depth we read, more easily we can answer the given questions. Okay, all right. Now let's start with the concept of plant physiology. Okay, so focus everyone. So in the chapter of a plant physiology, friends, what are the various topics that we have to cover? See, according to the 2025 NEET syllabus, we have to majorly focus on the first topic, photosynthesis, then cellular respiration. Correct, na? Cellular respiration. Next, forgot everyone, plant growth and development. Any other? No. So these are the 3 major topics that we can expect the questions. Okay, tell me how many number of questions that we can expect from this specific unit. Okay, an average, I am saying average. Okay, average I am talking about. You get 6 to 7 questions very easily. Average only I am talking about. So 6 to 7 questions means, friends, how many marks we can expect? 6 to 7 questions means 26 to 24 to 28 marks we can easily expect from this specific unit. Okay, undoubtedly it is one of the most important unit in the entire NEET syllabus, because some people can answer the questions even from human physiology also very easily. Some people cannot answer from the plant physiology. Why? Because questions will appear from applicative based, okay, statement based, assertion and reason, as well as the matching based also. Okay, so I am very sure whatever we are teaching here, the question never ever go beyond that level. The only thing you have to do is you have to get a complete knowledge and you have to apply that knowledge. Question never go beyond this level. You just focus on this very clearly. See, I'll teach biology in such a way that you no need to touch any book. I will only cover the entire chapter. I will only solve the MCQs. You have to sit in front of me and you have to practice. And you are, on other hand, you continue the botany and zoology as well. As a result, it becomes practice twice and we can answer any kind of a difficult question. Okay, now let's start with the most toughest topic, that is the photosynthesis. Now tell me, friends, see, according to our syllabus, these are the various subtopics that we have to cover. The definition of the photosynthesis, where the process of photosynthesis is taking place. Most importantly, the pigment involved in photosynthesis, cyclic and non-cyclic phosphorylation, very important, how ATP can be synthesized, which we can understood by chemiosmotic hypothesis, and dark reactions such as C3 pathway, C4 pathway, then CAM pathway, and factors affecting the process of photosynthesis. Okay, beyond that there is no other way of asking the question. Now let's try to understand everything very elaboratively. Okay, there are 150 slides are there. The question won't go beyond that. In case if you are feeling difficulty of reading the book also, just read the PPT once and directly solve the questions. It becomes very easy. Okay, even though you find a small doubt, please ask me. Okay, tell me, how can I define the word photosynthesis in very simple words? Photo represents light. Synthesis means what? To produce, to make something. What plants generally make? Food. Correct na? What is the major food that plants make? Glucose. Correct na? So photosynthesis is a process of production of food by using light. So every organism can perform the photosynthesis? No. Those organisms which are having the pigments that can absorb the light, only those organisms can perform the photosynthesis. Now look into the point. In order to perform the photosynthesis, plant require some raw materials. What are those raw materials? Sunlight, water. From where water is coming from? Soil. Water comes from soil, sunlight coming from the sun. It is the major source of the energy. So by using the water and sunlight, plants take carbon dioxide from atmosphere. Now by using these two, plant is reducing what? Reducing the carbon into sugar. Whenever plant preparing its own food, plant is going to release oxygen as their byproduct or waste product or end product. No plant purposely never ever release the oxygen. Ultimate goal of the plant is to produce the sugar. In this process it release the oxygen as a byproduct. Okay, which is the simplest definition. So look into the point. It is a physico-chemical process by which light energy or solar energy trapped by autotrophic organism to synthesize organic compound such as carbohydrates, which is sugar. Correct na? A very simple point. In this process of production of food, what they are releasing? They are releasing oxygen as their byproduct or the waste product also we can consider. Okay, now see the simple question that they asked in the exam. See here what I told you just now. In the photosynthesis we are reducing the carbon into glucose. Hence production of glucose is reduction reaction. Never confused with the statement. But whenever we are reducing the carbon into glucose, what is happening, friends? Here there is a breakdown of water, because upon breaking down of the water only there is a release of oxygen. Correct na? Hence we are breaking something which we call it as what? Oxidation. So how can you remember oxidation and reduction? I'll tell you in the upcoming slides. So if they ask you a question, there is a statement asked. Tell me, is it true or false? Slowly think and answer. The question asked is, production of glucose is a reduction reaction.
Yes.
Production of glucose is what reaction? Reduction. Breakdown of water is oxidation. Now the question is, photosynthesis is a redox reaction.
Yes, sir.
Correct na? Because reduction is happening and oxidation is happening. Suppose example in the exam, if they give a question, photosynthesis is oxidation reaction. No, that is not a completely right statement. It is a partially right statement. Sometimes in order to make us confused, they will say that photosynthesis is a reduction reaction, which is partly correct. Because ultimately we are producing sugar. But originally production of sugar is reduction, breakdown of water is oxidation. That's why photosynthesis is a redox reaction. Depending on the given options in the exam, we have to read that. Okay, now the question is very clear. You understand ultimate goal of the photosynthesis is what? Production of glucose. Now the question, in all our competitive exam, at least one or two questions from every concept comes from where that reaction is taking place. That is most common in the competitive exams. Now tell me where the photosynthesis takes place. Where photosynthesis takes place? In very simple, very good. Photosynthesis generally takes place in green parts of the plant. So such as, what are the green plants? Parts? Leaves, young stem and young green walls of the fruits. But compared to the young walls of the fruit and young stem, leaves are photosynthetically more active. That's the reason the process of photosynthesis exclusively happens in the leaf. Correct na? And where exactly photosynthesis happen? In a specialized organelle called as chloroplast. Chloroplast is a colored plastid or colorless? It is obviously a colored plastid. Why it is colored? Because it contains chlorophyll pigment. So in very simple words, whenever we use a word, it is colored. Always remember, it is involved in synthetic functions. In other words, it is involved in metabolic function. When I use a word, it is involved in the synthetic function. So chloroplast is involved in the production of what? Glucose. Correct, na? So undoubtedly we can say that all the chloroplasts are involved in synthetic function or metabolic function. Correct, na? And again there was a statement asked in the exam. Sometime chloroplasts are also involved in the storage function. Originally you read in the 10th standard also leucoplast. Yes. What are leucoplast? They are colorless. Correct, na? Amyloplast. What amyloplast will do? Store the starch. Correct, na? Proteinoplast stores the protein. But the question is here, you cannot say that leucoplast is involved in the synthetic activity. Why? Why? Because they do not have pigments. Forget about chlorophyll, they do not have any other pigment which is required for synthetic activity. Hence, they will be only involved in storage. Sometimes they will give a question, chloroplasts are also involved in storing something. We will discuss that. Chloroplasts are also involved in storing of, see here, storing of vitamins like E and K. That is a very important statement. Don't get confused over there. So, it is very clear the photosynthesis occur in the chloroplast. Now look into the beautiful structure of the chloroplast. If you observe the structure carefully, chloroplast is how many membranes are there? Two membrane, which is outer and inner. The space between the outer and inner called as intermembrane space. And if you look into the chloroplast very clearly, inside the chloroplast, what you people can observe, a flattened vesicle-like structure. Can you see that? Or flattened sac-like structure like this. This flattened sac-like structure we called as thylakoid. But surprisingly, all these thylakoids never ever arranged as a single. Most of the time, all the thylakoids are arranged in a sac or a group. So group of the thylakoids, what do we call it as? Granum. Correct, na? So all the thylakoids are arranged in a group called as granum. Now don't get confused. One group of thylakoids can be connected to the another group of the thylakoids via a thin thread-like structure you can see here. Correct, na? A thin thread-like structure. That thin thread-like structure, we call it as what? Lamella. It's not a lamellae, it is a lamella. And where it is located? Very, very good. This link located in the stroma. That's why we call it as? Stroma lamella. Very good. Stroma lamella. Widely called as fret channels. What is that? It is also called as what channels? Remember the name. Fret channels. Don't get confused. We call it as stroma lamella or fret channel. What else it contain? So it contain outer membrane, inner membrane, intermembrane space, flattened vesicle-like structure is called as thylakoid. Group of thylakoids called as granum. One group of thylakoids can be connected with the another by a thin thread-like structure called as lamella or fret channel. And inside the chloroplast, you can observe a fluid-filled structure or gel-like structure or matrix-like structure. You can call gel, you can call fluid, you can call matrix. Whenever I use a word fluid, matrix or gel, always remember they keep on moving. That's why we can call fluidized structure, which is called as stroma. Okay, and what else it contain? Chloroplast and mitochondria are said to be what? Self-replicating organelle or semi-autonomous organelle. Since they are having their own DNA and own ribosomes and RNAs also. And you read in the 10th standard very clearly, all the eukaryotic cells having the ribosomes and all the prokaryotic cells are also having ribosome. The prokaryotic ribosomes are and eukaryotics are ATS. But There are exceptions in chloroplast and mitochondria. Why? What is the reason? They are also contain what ribosome? 70S. My question is why? We will read in the evolution. According to the endosymbiotic theory, it was clearly proven that chloroplast and mitochondria derived from bacteria. That's the reason the ribosomes are also 70S. And what else the chloroplast contain? DNA. The DNA, again the question in the exam, what is the shape of the DNA in the chloroplast? Which is double-stranded circular. Very, very good. Okay. And again the question, what is the most important protein coded by the chloroplast DNA? What is the protein? The protein coded by the chloroplast DNA is what? Rubisco. Rubisco is said to be the most abundant protein on this planet Earth. In the human body, what is the most abundant protein in the human body or in the animal kingdom? Collagen. Why collagen is most abundant? As the word itself indicating, abundant, present at many areas. It is present in tendon, ligament, joints, arteries, uterus, everywhere. So when I say Rubisco, Rubisco is also abundant. Why? If you take any leaf, 40% of the leaf contain Rubisco. Hence, every plant, every leaf of every plant on this earth contain Rubisco. That's why it is most abundant. And that Rubisco can be coded by a chloroplast DNA. What else it contain? Chloroplast contain, can you see the picture? It contains lipid droplets and it contains starch granule. What is the interesting point? Lipid droplets are small in size, but more in number. Starch granules are large in size, less in number. In other words, chloroplast can store the lipid also as well as starch also. This is the simple outer structure what you can see, right? And it is also important to understand the inner view of the chloroplast. In other words, the microanatomy of the chloroplast. Why we have to read the microanatomy? We already read a very simple structure of the chloroplast. Because in the photosynthesis, variety of reactions happen at different locations. Hence, it is important to understand the entire structure. Now tell me all of you, what is this? Thylakoid, a flattened vesicle-like structure. Now look into the point. Now what is this? Granum. Very good. Now if you observe the membrane of the thylakoid, what is that? Membrane. See, this is the thylakoid having a outer membrane, correct na? This is the membrane. So the membrane of this particular thylakoid, don't get confused, we call it as lamellae. Okay, it is not lamella. What is lamella? One group of thylakoids can be connected with another group. This is called lamella. And it is what lamella? Stroma. Because it is protruded or moved towards the direction of stroma. So this is what? Thylakoid lamellae. Correct na? In simple words, we can call thylakoid membrane. But they don't use the word membrane in the exam. They use the word thylakoid lamellae. Correct na? Now remember, in any organ, whether it is a human organ or a plant organ, inner space, we always call it as lumen. Okay. So why we have to read this? Because different reactions happen at different places. Correct na? So now anyway, you can read it. The PPT designed in such a way that every single statement will help you to recall everything. So next time I won't read all this, but you can read it easily. Chloroplast is what organelle? Double membrane. And where these chloroplasts are found? In a leaf. In a specialized cells called as mesophyll cell. Now what we discussed, how many membranes are there? Outer and inner. Again the question which we can get in the exam, which membrane is more permeable? Outer membrane is more permeable compared to the inner membrane. And the space is called as, what is this? Periplastidal space or intermembrane space. And matrix or gel-like structure called as stroma. Correct na? And what are thylakoids? Flattened vesicle-like structures called as thylakoids. Correct na? Now most important one group of thylakoids can be connected with other by channels called as stromal, lamellae or fret channels. Correct na? No need to get confused. Since it contains the DNA, we call it as what? Semi-autonomous organelle. No need to get confused. So, the entire process of photosynthesis, whatever we are going to discuss, everything summarized in this slide. If you understand this slide, almost all concept will be very clear to all of you. Tell me, what is the ultimate goal of the photosynthesis? Production of glucose. This is the ultimate goal. Let's consider I am a plant. My ultimate job is to produce glucose, but unfortunately I cannot produce the glucose very easily. If I want to produce the glucose, I require abundant amount of energy first. First I will produce energy. If I have energy with me, I can produce the glucose very easily. Anyway, how we produce the glucose we studied na? By reducing carbon. Correct na? This is more important. You always remember production of glucose is reduction. So I want to reduce the carbon into glucose, by that I can produce glucose anyway. But if I want to reduce the carbon, what I require? Energy. What are the energies I required? ATP and what is the full form of ATP? Adenosine triphosphate. It is said to be the universal energy currency of the cell. Whether it's a plant, animal, yeast, fungi, bacteria, anything, it requires ATP. Now, what is NADPH? Nicotinamide adenine dinucleotide phosphate. Nicotinamide adenine phosphate. So this is also energy only, okay, but indirect. So instead of calling it as the energy equivalent, what do we call it as? Why do we use the word reducing equivalent? Because it is undergoing reduction. You're getting the point. And mostly you remember, we will read in the cellular respiration. Whenever you heard the name of NADPH and FADH2, remember a common statement, they are acting like reducing equivalent. Most importantly, they are acting like electron carriers. Never forget these two statements. Both are reducing equivalent, NADPH and FADH, and electron carriers, which means they transfer electron from one to other. So our ultimate goal of the photosynthesis is production of glucose. Before that we have to produce energy. But if we want to produce the energy, what do we require? We require light. Without light it is impossible to produce the energy. But our goal is not to produce energy, right? Our goal is to produce glucose by reducing the carbon. In order to reduce the carbon into glucose, there is no requirement of light. What is required? Energy. But to produce energy we require light. That's why photosynthesis is broadly categorized into light reaction and dark reaction. No point of confusing, since I am using the word light reaction, which means compulsory they require the presence of light. So it only occurs in the daytime. Correct or not? Correct na? Now, dark reaction. Dark reaction doesn't mean that they are occurring in the dark. Dark reaction means that they are light independent reaction. What do you mean by that? See, I am a plant. I want to prepare the food daytime also. I want to prepare the food night time also. So whether the light is present or absent doesn't matter. What is matters? Energy is present or absent. That's why dark reactions are said to be light independent. When they occur, night or day? When they occur? Just now I told you, no, you are all plants. You want to prepare food daytime also, night time also. Then dark reaction occur both day and night. They are light independent. Light is present or absent doesn't make any difference. Energy is present or absent, that makes the difference. You're getting? So our entire lesson is we have to read extreme depth of light reaction, extreme depth of dark reaction. Okay. But now the first question, which is commonly asked in your exams. Where is the site of light reactions? Why? And see, light reactions are see, what is this? What is this? Thylakoid. What is this? Thylakoid membrane, also called as lamellae. So light reactions exclusively happens on the lamellae. Thylakoid membrane or lamellae. Now you have to ask why. Why it is happening only on the thylakoid membrane? This is the fixed picture of thylakoid membrane. membrane made up of lipids, right? Now why it is happening only here? Because all the photosystem, ATP synthase enzyme, all the electron carriers present on the thylakoid membrane. That's why light reactions are happening on the thylakoid lamellae or membrane. Now the question is very clear, where the dark reactions taking place? Dark reactions are exclusively taking place in the stroma, and the question is why? Because if dark reactions wants to occur, we require a lot of enzymes. An example we will discuss in the upcoming slide, Rubisco, pep carboxylase enzyme. All the enzymes are present in the stroma. That's why dark reactions such as C3, C4, CAM pathways occurs in the stroma. But to produce the energy, all the reactions happening in the thylakoid membrane or lamellae. No need to get confused over there. Okay. Now see this question in NEET 2025. In photosynthesis, light independent. Why we use the word independent? Because we use the word independent? It doesn't make any difference whether it is light is present or absent. Light independent reactions takes place at photosystem 2, stromal matrix, thylakoid lumen, photosystem. Sometimes they can call gel structure. Okay. Sometimes they can call fluidized structure. Okay. Anything is same. Obviously it occurs there. Okay. Now don't get confused. Now tell me, what is the ultimate goal of it? Is a very broad concept. Okay, you have to read minimum 3 times or 4 times, then you can answer any kind of a question that comes in the exam. We'll take it as a challenge. Okay, now light reactions are taking place where? Thylakoid membrane or lamellae. Now the first question you have to remember, goal of light reaction. Never forget this until we synthesize these two, ATP and NADPH. This is our ultimate goal. Now question is very clear, friends. In the light reactions we require light mandatorily, correct na? That's why we call light reaction. Then in the plant, who will absorb the light? Because my goal is what? I have to absorb the light energy, I have to convert into chemical energy. By that I can produce ATP and NADPH. So someone has to absorb the light. Who is going to absorb? Pigments, correct na? Not only chlorophyll, forget. So generally pigments are the one which can absorb the light. Now all the pigments present in the plant body broadly categorized into principal pigments and accessory pigments. Sometimes instead of using the word accessory, they use the word antenna. Anything is same. They will call antenna or accessory pigments. Now look into the point. Principal pigments are broadly categorized into many types. Accessory also many types. Now if I ask you a question, which will be more in number, or forget about the number, which are more important? More. Principal pigments are more important. It doesn't mean that we do not require accessory or antenna. Both are required. Plant require all. But most important is what? Principal pigments. What is, you have to remember this till the end. In the plant, the most major principal pigment is chlorophyll A. It is exclusively present in the green plants. So chlorophyll A is the one which is generally absorbing the light in the green plants, higher green plants. Let's consider angiosperms, higher green plants. Now see here, this is normal chlorophyll A which is present in the green plants. Now I am using a word bacterial chlorophyll A and chlorobium chlorophyll. What is the difference between these three? Don't get confused. The same chlorophyll A, in other words, modified chlorophyll A present in the purple bacteria, call it as bacteriochlorophyll A. When I use a word, sir, what is meaning of modified? Structurally modified. Similarly, the structurally modified chlorophyll A present in the green sulfur bacteria, call it as chlorobium chlorophyll. No point of confusing. So these two are also chlorophylls only, but they are structurally modified. We'll see what is their structure actually. Now these are all principal. When we use the word principal, which is major, correct na? More important. The next, accessory or antenna. Now see here, they are chlorophyll B, C, D. What is the interesting point? Chlorophyll B, C, D are not present in the higher green plants. Then where they are present in the lower plants such as green algae. Don't confuse, very important. Next, carotenoids. Carotenoids are broadly categorized into two types. Number one, we called carotenes. What is that? Next one, xanthophyll. Now when you look into the structure of the carotene, I will show you in the next slide, it just contains carbon and hydrogen, that's all, nothing else. It contains carbon, hydrogen, we'll see that. But if you look into the structure of the xanthophyll, it also contains carbon, hydrogen, along with that it is also contain oxygen. That's the simplest difference. Now, carotenes where they are present? Plants, algae and bacteria. Xanthophyll also present everywhere. Now the question, give me some example of carotenes. Beta carotene, where it is present? Carrot. Carrot is orange color because of the beta carotene. And lycopene, which is present in the tomato. Tomato is red in color because of the lycopene. Very simple. Now next one, phycobilins. Example of the xanthophyll, lutein. What is that? Lutein. You have to remember. Okay. Next, phycobilins. Exclusively present in cyanobacteria and rhodophyta. Examples are phycoerythrin, phycocyanin and L-phycocyanin. What is the question asked in the exams? The question asked, among all the photosynthetic pigments which are water soluble. Remember, you will understand why it is. Chlorophylls, carotenoids are strictly lipid soluble. We will see why. And phycobilins are the only pigments which are water soluble. Now let's understand why they are lipid soluble and why they are water soluble. Very important. Okay. Now look into the picture. Okay. There are 7 different types of chlorophyll pigments are present in the autotrophic organism. Look into that: chlorophyll A, B, C, D, E. And can you see that? Bacteriochlorophyll and bacterioviridin. Just now we discussed bacteriochlorophyll and bacterioviridin. These are all the type of modified chlorophyll A, but not present in the plants. So where they will be present? In the bacteria. You are getting the point. Whereas chlorophyll A to E, they are found in plant kingdom. But when I say higher plants, what the higher plants contain? Only chlorophyll A. You are getting. Remaining B, C, D, E, where they are present? In the lower plants such as green algae. Okay, don't get confused over there. Okay. Now what are carotenes? Chlorophylls are also fat soluble. I'll explain you why. Carotenes are also fat soluble. Correct na? Now they absorb the light energy in blue violet region of the spectrum and transfer the absorbed energy to where? Why they are transferring to chlorophyll A? Because we said na, chlorophyll A is principal pigment. That's why they will transfer to that only. Okay. Now next one, xanthophyll. See, they have said that it is fat soluble pigment, but it is less fat soluble, more water soluble. Don't get, sorry, I'm so sorry. This is xanthophyll. Xanthophyll is fat soluble only because xanthophyll is a type of the carotene only. That is also a fat soluble. The next step, phycobilins. These ones, they are extremely water soluble. Now let's see how. Now all of you look into the picture. Again, there is a great possibility you people expect the questions from the structure. You no need to remember the structure. You have to remember the structural feature. How does it look like? Now when you observe the structure of the chlorophyll, remember chlorophyll having head, chlorophyll having tail. The head of the chlorophyll, what is that? Anyone remember? The head of the chlorophyll called as a porphyrin head. The tail of the chlorophyll we called phytol chain. If you ask me how to define a phytol chain, simply I will say that it contains hydrogen carbon. That's all. A long hydrogen carbon chain called as phytol chain. Simple. Then how can I define porphyrin head? All of you look into the picture. In the head, what you people can observe? A, B, C, D. They are called pyrrole rings. What? Pyrrole. Now how many pyrroles are there? A, B, C, D. 4. 4 pyrols called as tetrapyrrole. A tetrapyrrole ring coming close together and surrounding the central metal atom magnesium. How they are surrounding the central metal atom magnesium? Via nitrogen. Can you see the picture? So that is called porphyrin head. What is porphyrin head? A tetrapyrrole ring come close together and surround the central metal atom magnesium. Correct na? Which is called as porphyrin. Now from the starting of the lesson we are saying that chlorophyll molecule absorbs the light. How? Chlorophyll molecule in the center, what it is having? Magnesium. What is the function of magnesium? Magnesium act like a chromophore. What is that? Chromophore. This is also a question. Chromo, color. Phore, which absorbs. This is the one which absorbs the color. That's why chlorophyll absorbs the light. Okay, no need to get confused over there. Now, so it is containing what chain? Long hydrocarbon chain. Hydrocarbon chain means what? Lipid soluble. Now look into the structure of carotenoids. No confusion. In simple, I have written everything. Carotenoid said to be isoprene unit. What is that? Isoprene. What is the isoprene here? Don't get confused. It is a 5 carbon containing compound with alternate single bond, double bond, single bond, double bond, single bond, double bond, single bond, double bond. 5 carbon containing compound with alternate single bond and double bond called as isoprene unit. You are getting? Now, what is this? Bilin pigment. Bilin pigment also contain pyrrole. 1, 2, 3, 4. So it is also called as tetrapyrrole. Where we heard the tetrapyrrole just now here. What's the difference? Here tetrapyrrole is circular. Now here linear. So that's the structure. Linear tetrapyrrole is called as bilin pigment. 5 carbon containing compound with alternate single bond, double bond called as, which is called isoprene unit. Porphyrin head and phytol chain is called as chlorophyll. Now what is the common point? In the chlorophyll we have long hydrocarbon chain. Correct na? In the carotenoid we are having long hydrocarbon chain. You can't see the long hydrocarbon chain in the bilin. Hence they are water soluble. Wherever you can see hydrocarbons, they are lipid soluble. No point of confusion over there. Okay. So the chlorophyll formula, chlorophyll A, C55H72O5N4. You have seen where 4 nitrogens are present? Tetrapyrrole. Tetrapyrrole surrounding magnesium with the help of nitrogen. Never confuse. In case of chlorophyll A, 5 oxygens are there. Here there is a presence of 6 oxygen. Here 72 hydrogens and here 70 hydrogen. That's all the difference. Okay. Now the question is very clear. If I ask you a question to all of you, just now we discussed we are having chlorophyll A, B, C, D, E. Correct na? Now this is also a question. Now we have chlorophyll A, B, C, D, E. Every chlorophyll having porphyrin head and phytol chain. Now question is here, how can we differentiate one chlorophyll molecule with other chlorophyll molecule since entire structure is same? How can you differentiate? Based on oxygen and position of the carbon. Very good. All are correct. The major difference is based on side chain. What? Based on side chain. If you observe the structure of the chlorophyll A, side chain it is having methyl group. If you observe the chlorophyll B, having aldehyde group. Bacteriochlorophyll A having something else. So one chlorophyll molecule can be differ with the another chlorophyll molecule with the help of what? Side chain. No point of getting confused over there. Chlorophyll A having methyl group. Correct na? Chlorophyll B having aldehyde group. But what is the common? Both chlorophyll A and B contain magnesium. Where it is present? In the center of the tetrapyrrole, which is called porphyrin ring. Correct na? Now listen, the lesson is very lengthy, very interesting if you understand. If you don't understand, this is the toughest unit. Okay, focus. What is our goal of light reactions? Production of ATP and NADPH. Don't forget this. If we want to reach this level, we have to read so much first. Now, what is the function of the pigments? Absorption of light. Now, there is a simple question to all of you, think properly. So, now chlorophyll absorb the light, carotenoids absorb the light, bilin pigments also absorb the light. Then what is the requirement of all the pigments in the plant? Any one is enough now, because all are absorbing the light only. What is the reason? All are absorbing the light. Very good. What do you mean by that? Different pigments absorb the light at a different wavelength. First difference. Second and most important difference, this is also they will raise the question. Which of the following photosynthetic pigment protect the plant from photooxidative damage? Which is? Carotenoid. What is called photooxidative damage? High intensity of sunlight. See, we are animals, when we expose to the high intensity of sunlight, we will run away because we can escape, because we can move. But plants cannot move. They have to tolerate that. How they can tolerate? By using carotenoids. Okay, they are said to be photoprotective pigments. Okay, so no need to get confused over there. Okay, it is photosynthesis. Okay, now what is the common function of every pigment? Absorption of light. So based on the absorption of light, there were two spectra were derived. Absorption spectra, action spectra. Okay, I'll slowly try to understand, it is more interesting. When I use the word absorption spectra, listen, very simple point. Absorption spectra is talking about pigment. Action spectra is talking about process. So now let me define you the absorption spectra in a very simple way. At which particular wavelength a pigment absorbs the light? What? At which particular wavelength a pigment absorbs the light? That is absorption spectra. Now let me explain you more elaboratively. Don't get confused, to try to focus on the picture correctly. On the x-axis, what we have taken? Wavelength of light. On the y-axis, I have taken amount of the light absorbed by those pigments. Now look into the point. What are the various pigments I have taken? I have taken chlorophyll A, chlorophyll B and carotenoids. Focus on the picture. Now all of you remember one common statement. Whatever the, see, I am a pigment. Whatever the color absorbed by me, I never appear in that color. Whatever the color reflected by me, I will always appear in that color. Whatever the color absorbed, it never appear. Whatever the color opposed or reflected, it appears in the same color. Now look into the point. Let's take the number one, chlorophyll A. Look into the point. When you look into the structure of the chlorophyll A, focus on the picture. Chlorophyll A, see here, this is dark blue light, green light, yellow light, orange light, red light. Now, if you observe the picture of the chlorophyll A, friends, see this. This is, the peak is extremely high till here, correct na? After that peak was fallen down. So what you have to observe, what you have to understand by that? What happened? So look into the point. So can I say chlorophyll A absorb dark blue light easily? Because peak is very high. It absorb light blue light also very easily. But can you see it absorb green light? No. So it is fallen down. So it is reflecting or absorbing the green light? Reflecting. Since it is reflecting, it appears in green. So why plants appears green in color? Because chlorophyll reflects green color. Now look into the carotenoids. Carotenoid absorb dark blue light, correct na? Light blue light, it is also absorb green light. Will it absorb yellow light? No. Orange light? No. Red light? No. Why carrot is orange color? Because it contains carotene, which reflect orange light. Why tomato is red in color? It contains carotenoid named as lycopene. It reflects red color. Correct na? So whatever the color absorbed by the pigment never appears in that color. Whatever the color reflected by the pigment, it appears in that specific color. No point of getting confused over there. So what the absorption spectra is explaining us, look into the definition now. The absorption of a light at different wavelengths by different pigments named as absorption spectra. So it is only talking about a pigment, correct na? Now when I use the word action spectra, it is not at all talking about the pigment. It is talking about process, which means at which wavelength a particular process occur maximum. In simple words, you take any process as an example. Let's consider photosynthesis only as a process. Tell me what is the most suitable wavelength for the photosynthesis? The most suitable wavelength will be 600 to 700 nanometer. Why? Because at this wavelength, all the pigments absorb maximum amount of light. So what is action spectra is talking about now? At which wavelength a particular process occur. So the rate of photosynthesis is measured in terms of what? Oxygen release at different wavelengths of light is called as action spectra. Okay, every process do not have the same action spectra. Every process having a different action spectra. In simple words, absorption spectra talks about pigment, action spectra talks about process. No point of getting confused over there. Okay, let's look into the questions quickly. It's a very broad unit. Imagine 157 slide means we have to read extreme depth, and how difficult they may give the question. If they give the question beyond the level of 2025 paper also, you can answer so easily. Okay, now we already read this. What we read? Light and dark reaction. What is the ultimate goal of the light reaction? Production of ATP and NADPH. Correct na? So always remember friends, never get confused. You will understand in the upcoming slide. In the light reactions, there is a breakdown of water, which means there is a release of oxygen. So question is clear. In the dark reaction, breakdown of water never occurs. Oxygen release also never occur. So production of ATP and NADPH also never occur in the dark reaction. Utilization of ATP and NADPH occur. You are getting the point. But production occur in the light reaction. Breakdown of water also occur in the light reaction. Oxygen release also occurred in the light reactions only. ATP and NADPH are what? Energies. Together we call it them as the assimilatory power. Okay. Now look into the point very correctly. Now quickly answer the question without having a confusion. What is the question? Which of the following is not a product of light reactions of photosynthesis? NADPH we will produce, right? ATP we will produce. When water molecule break down, there is a release of oxygen. So the only product which never produced is the NADH. Correct na? No need to get confused over there. We'll read every single question up to 2025. You can answer every question so easily. Okay, friends, now from now onwards you focus. It is becoming little tough now. Okay, so next half an hour is little bit difficult and very much confusing. Just focus on the picture so clearly and mandatorily revise whatever I discussed today. By that tomorrow it becomes easy. By the end of the topic I will only solve 100 questions. By that you become perfect. Light reactions of photosynthesis. So can you see the direct question, which is the NCERT line? Light reaction or photochemical phase includes? Why we use the word light absorption? Because pigments will absorb the light. Splitting of water, release of oxygen, production of ATP and NAD. All are happening in the light reaction. They will ask a direct question, which of the following event is not a part of the light reaction? Correct na? Where it is happening? Entire light reactions are happening on thylakoid membrane or lamellae. Correct na? Now tell me why? Why only happening on the thylakoid membrane? Because all the photosystems are present over there. How many photosystems are there? Two photosystems. Very, very, very important here. How many are there? Two. Now what are the names of the photosystems? Photosystem 1 and photosystem 2. Can anyone give me the idea why we call photosystem 1 and 2? How we have given the name? Based on? It is not at all based on the wavelength absorbed. It is just based on the series of discovered. First discovered, second discovered. That's all. There is no, based on, not based on the wavelength. Now look into the most important statement. This is thylakoid membrane. Correct na? See, don't confuse, I'll explain in the upcoming slides elaboratively. This is the thylakoid membrane. This is what we can call photosystem. Correct, na? Inside the photosystem, you will read in the upcoming slide, there is something called as LHCs, light harvesting complex. Now it is very clear. So every photosystem contains LHC. Every LHC made up of two components. What are those? Reaction center, antenna, pigments. These are all common. Reaction center is made up of what? Chlorophyll A. Just now we discussed in a principal pigment, also called as reaction center. Reaction center is always made up of A. It is fixed. Then antenna pigments are also called as accessory pigments, made up of chlorophyll B. Very good. You can say C, D, E, xanthophylls and carotenoid. These are, never forget this line till the end of the lesson. Antenna pigments except chlorophyll A, remaining all are antenna pigments. Now friends, don't confuse, you will understand in the upcoming slides elaboratively. Photosystem 2, photosystem 1. So undoubtedly both photosystems contain reaction center, correct na? So reaction center is exclusively made up of chlorophyll A. So which means in the photosystem 1 we have a chlorophyll A. Photosystem 2 also we have chlorophyll A. Correct na? Now why we called photosystem 2 as the P680? Why we called photosystem 1 as the P700? Because whatever the chlorophyll A that is present in the reaction center of the photosystem 1 absorb maximum amount of the light at 700 nanometer. That's why we can call P700. Whereas same chlorophyll A absorb the light at 680 nanometer. That's why we call it as P680. Correct na? No confusions at all. Now you people look at the diagram and tell me what are all this. Consider this is the exact question asked in your exam. What is this membrane?
Thylakoid membrane.
Very good. And what is this entire photosystem? Inside the photosystem what we have? LHC. Okay. LHC made up of and antenna pigment. Can you see this picture, friends? This is called as a reaction center. Remaining what else it contain? Antenna pigment. So these are all antenna pigments. What you people can observe, ultimately antenna pigments absorb the light. Can you see that? Finally sending the energy to where? Reaction center role. Never ever get confused over that. Okay. Now listen. So light harvesting complexes, the photosynthetic pigments and several protein complexes. What are those? Photosynthetic pigments and protein complexes are together arranged into light harvesting complex where they are present within 1 and photosystem 2. Correct na? So first thylakoid membrane, on the membrane, inside the photosystem, inside the LHC, pigments, which are reaction center and antenna pigments. Correct na? No need to get confused. Okay, each photosystem has all the pigments forming LHC, which are also known as antenna molecule, also called as accessory pigments. Ultimate goal is what? Absorption of light. No point of getting confused over there. Now let's give me one moment of time. Okay, now every student has to understand the lesson very correctly from now onwards. What is the ultimate goal there? So where the reactions are happening? Thylakoid membrane. Correct na? Now look into the point. This is the direct NCERT picture. So you tell me, friends, what is this dark green color which is separated from others? Reaction center. What is all this light green color? Antenna pigments made up of chlorophyll B, xanthophyll, carotenoid. Simple. What is our ultimate goal? Production of energy. For that, all the pigments will absorb light. No, never get confused. Now, all the antenna pigments are absorbing the light. Can you see that? Absorbing the light and passing the energy to where?
Reaction center.
Now the reaction center, upon absorbing great amount of energy, it get excited because it got a lot of energy. Upon absorbing the energy, it get excited and release excited electron. So now electron is released out, someone has to accept this electron. So whomever accepting the electron, we called electron acceptor. How many electron acceptors are there? There are many. See, let's consider I am reaction center. You 4 people are electron acceptor. So first I will send electron to you, then you will pass to others to other. So let's consider 1, 2, 3, 4. Electron is passing from here to here, here to here, and here to here. Whenever electron is moving from one place to the another place, it lost energy. What?
Lost energy.
That energy can be used to produce?
ATP and NAD.
No, only ATP. Okay, so let's see, let's understand more elaboratively how exactly it happens. So the production of ATP called as what? Phosphorylation. What? So understand, use the brain, apply the knowledge. What is called production of ATP, friends? ADP combining with
Phosphate.
inorganic phosphate. Then we get ATP. Now you only tell me, can I add ADP with PA that much easily? No. It is very much energy requiring process. From where that energy is coming from? Just now we discussed when electron is moving from one electron acceptor to another electron acceptor, there is a loss of that energy. They will take it and produce ATP. Let's understand more depth. So production of ATP called as phosphorylation. Undoubtedly energy comes from light, because light is the ultimate source of energy. So that's why it is called
Photophosphorylation.
So photophosphorylation is the production of ATP. So based on how these electrons are moving, we categorized the photophosphorylation into cyclic and non-cyclic phosphorylation. No point of confusing over there. Why did I use the word cyclic? Suppose electron started moving here. Again it comes back to the same place. Suppose electron started here, it is going somewhere. That's why it is called non-cyclic. And most important, you have to remember for the exam, cyclic photophosphorylation never ever happens in the plants. Okay, never ever confuse. Cyclic photophosphorylation occur in the bacteria. Non-cyclic phosphorylation strictly happens in the plants. And the most important statement for the exam, cyclic phosphorylation require only one photosystem. You got the point? Whereas non-cyclic require both the photosystems. We'll discuss more depth. Look into the point now. Now again, tell me all, what are all these? Antenna pigments are accessory pigments. Very good. And what is this? Very good. So they absorb the light, correct na? One absorb the light and transferring to other. So I want you to remember a small definition of oxidation and reduction. Tell me in chemistry, how can you remember oxidation and reduction? Simple words. Loss of call it as
Oxidation.
so simple. Remember a shortcut named as oil rig. Oil rig. Okay. O represents,
Oxidation.
L represents losing. Losing of electron is oxidation. R is reduction. Gaining of electron is called as reduction. Always remember like this. Okay. Simple oil rig. Now look into the point. Focus all of you. First, let's consider this is first antenna pigment. Second, third, like that. So first antenna pigment gain the electron. Correct na? So it undergone reduction or oxidation? Reduction. It gained it, right? But it won't keep electron with it. What it will do? Upon transferring, it becomes oxidized. And second one get reduced. Correct na? So series of oxidation, reduction reactions are happening. Correct or not? Now this kind of a transfer of energy from one antenna pigment to the another antenna pigment called as resonance transfer of energy. What is that? Resonance transfer of energy. That is also a question where a series of redox reactions are taking place. Now friends, let's consider what we are discussing. Cyclic photophosphorylation. Never get confused. Now all the antenna pigments absorbed the light and send the electron to reaction center. What happened to the reaction center now? Get excited, released excited electron. Upon releasing the excited electron, someone has to accept it. Now the question is this: who is the first electron acceptor in the, this is the exact question. Okay, who is the number one electron acceptor? FD, ferredoxin. Then PQ, plastoquinone. There are two type of plastoquinones are there, quinone A and quinone B. Then cytochrome B6F. Then very important one, PC, plastocyanin. Then finally electron is coming to where? Reaction center. Since all the electrons are moving in a cyclic manner, we called as cyclic photophosphorylation. Now look into this, what's happening here? Whenever electron is moving from one electron acceptor to the another electron acceptor, what's happening? There is a loss of energy. See, you are moving from here to there, obviously you will gain or lose? You will lose the energy. Then that energy can be used by ADP to combine with PI. Now you will ask me a question, sir, from where this came from? They are all present inside the cell, na? What is A represents? Adenine. Adenine is what? Base, nitrogenous base. They are already present inside. Correct, na? Sugar present inside. Phosphate present inside. They are not coming from outside. They are already present inside, but they are joining. Then there is a production of ATP. Now all of you slowly think and answer the question. Do we really fulfilled the goal of light reactions by using cyclic photophosphorylation or not?
No.
Why? What is the goal of light reaction? Production of ATP and NAD. This is the question asked more than 5 times. Okay, in the cyclic photophosphorylation we produce only ATP, that is which equivalent? Energy equivalent, which we have not produced, reducing equivalent. Correct, na? What is the reason? Simple reason, there is only one photosystem is present. Okay, and where this cyclic is happening? In the bacteria. Never confuse, it is happening only in the bacteria. See, 90% you should always compare between cyclic and non-cyclic. Okay, most of the questions will appear from the non-cyclic. So where the non-cyclic is happening? Plants. So I never encourage you to compare between cyclic and non-cyclic, but to remember easily you compare. Okay, which is the first electron acceptor in the cyclic? Cyclic. First electron acceptor, FD, plastoquinone, F and plastocyanin. Clear. Again it come back to the origin. Now in case of non-cyclic, how many are there? 2 photosystems are there. See photosynthesis topic we have to connect extreme depth, then you can answer 2 questions. If you read just on a glance, you can't understand the question also. Okay. Now look into this. How many are there? P680 and
700.
very clear. Now look into the question properly. This is what, friends? Antenna pigments. This is wonderful. Okay. Now antenna pigments absorb the light, send to the, then reaction center get excited, release the electron. Now again the first question asked in the NEET exam, which is the first electron acceptor from P680, which is a pheophytin. What is that? Pheophytin. Then what is this? Quinone A, quinone B, same. Then cytochrome B6F complex, same. Then plastocyanin, same. The only difference is what here? Here FD, PQ, cytochrome B6F, plastocyanin. Here pheophytin, plastoquinone, cytochrome B6F and PC. But you should never compare. This is happening where? Bacteria. This is happening in the plant. To remember, I am explaining you, that's all. Okay, never compare. But here what happened, friends? From the plastocyanin, where the electron came back? To the reaction center. But it never happens like this. Can you see the picture now? From the plastocyanin, electron transferred to? Very good. From the P700 again? Wonderful. Now here is the question. Okay, never get confused. Now, who is the first electron acceptor? You can't remember that much easily, you have to read it. Who is the electron acceptor? A naught. Next, A1. See, I'll tell you, A naught nothing but modified chlorophyll A. It is called A naught. A1, it is having a name, you don't get that question. A1 called phylloquinone. That is also type of chlorophyllol. Then after A naught, A1, series of iron sulfur proteins. FESX, FESA, FESB, FD, FNR. Can you see that? So from PC, electron reached P700 to the reaction center, A0, A1, iron sulfur X, A, B, FD, FNR. Correct na? Then there is a production of NADPH. Now answer the question which is asked in this 2025 MCET question paper also. Now first answer, which is the electron donor for the P700? Which is the electron donor? PC, which is plastocyanin. Which is the primary electron acceptor from the P700? Primary electron acceptor is what? A0. And most important one, slowly think and answer. Which is the last electron acceptor that is involved in the production of NADPH? Slowly think. What is my question? Which is the last electron acceptor involved in the production of NADPH? What is the answer? See, 90, not 99, everyone will choose the wrong answer. See, after FNR, where the electron is coming? This is the exact question. It comes to NADP plus. Later, there is a production of NADPH. Correct, na? No point of getting confused over there. Okay, this is easy. You understand. Now, here, what is the challenging part? You have to remember. Okay. Now, this is a very interesting point. How many photosystems are there?
2.
How do you know there are 2 photosystems? See, in order to understand this, there was a wonderful experiment was proposed. Experiment was done by a great scientist named as Emerson. You read na? Okay. What he did, no need to get confused here. See, understand presence of 2 photosystems. See the line operating. What do you mean by that? P680 absorb the light. At the same time, P700 also absorb the light. So, if the process of photosynthesis wants to occur, it is mandatory that photosystem 1 is also required, photosystem 2 is also required. You are getting my point. Now, the interesting point, what they have done. Okay, now do not confuse with my statements. You just understand for your better remembering. We have P680 and
P700.
correct na? Always remember, friends, P680 absorb shorter wavelength. Okay, when I use the word shorter wavelength, always remember red light. What is that? Red light. P700 absorbs longer wavelength, which is far red light. You're getting the point. Red light and far red light. We will read in the phytochromes. Okay. Now, what scientist does first? First, they have exposed, first they did this experiment in the chlorella. That is green algae. They have taken the chlorella, that is green algae. They exposed the chlorella to the monochromatic light. What is monochromatic light? Only one type of a light. You're getting the point. Let's consider I am a bacteria. You all people exposed me towards a monochromatic light, that is only single light. That is which light? P680. I can say red light. You all exposed me towards the red light. What you people observed? There is a complete decline in the photosynthesis process beyond this P680 nanometers. You're getting my point? First, I am a bacteria. First, you have exposed me to P680. That is red light. Forget about P680. You have exposed me to the red light. So, beyond 680 nanometers, in my body, the rate of photosynthesis dropped down. Later, what they have done, they have taken me again, chlorella. This time, they have exposed it to far red light, which is 700 nanometer. Above 700 nanometers also, photosynthesis is drastically dropping down. Later, what they have done, they have taken me again. This time, they have exposed me to the P680 and P700 at a time. Then, rate of photosynthesis is always increasing. What do you mean by that? Which means that in the plant, 2 photosystems are compulsory required for the process of photosynthesis to occur. You are getting the point. Not one photosystem. But in bacteria, there is only one photosystem. And so, P680, that is PS2, absorb shorter wavelength. P700 absorb far red light. So what is the question? If the process of photosynthesis wants to occur, we require red light also, we require the far red light also. So initially when the exposure only to the 680 red light, what is happening? Above P680, what happened? The photosynthesis was completely declined. That is called red drop effect. Correct na? So many times asked question. See this? Emerson noticed that sudden drop in the rate of beyond 680 nanometer. In which region?
Red region.
So which photosystem? P680. Then he identified already one photosystem is existing, but photosynthesis is dropping down. Then later they noticed that drop in the photosynthetic rate beyond red region spectrum can be restored by providing shorter and longer wavelength. What is that longer wavelength? Far red light. Far red light is absorbed by P700. Correct na? So only red light, there is a drop in the photosynthesis. We call, when I add the longer wavelength, photosynthesis rate what? Increase. That is called Emerson enhancement effect. What this proved us? This proved us if the process of photosynthesis wants to occur, requirement of two photosystem, and two photosystems has to work simultaneously or sequentially? Simultaneously. Answer the question. Operating simultaneously. Correct na? So questions will come in a very simple manner if you have a good knowledge. Okay, otherwise everything seems to be very difficult. Okay, now let's we understand non-cyclic very clearly, and we also proved just now two photosystems are mandatorily required, and we also read that two photosystems work simultaneously, which means P700 is absorbing the light, P680 also absorbing the light. Correct na? Now think answer. P700 absorb the light, send the electron to where? Reaction center. Correct na? Now can I say P700 antenna pigments are deficient of electron?
Yes, sir.
Correct na? Now someone has to replace this electron. Who is replacing? Plastocyte. Wonderful. Now again, P680 is also absorbing the light, sending the electron to reaction center, which means P680 is also deficient of light, so electron. Correct na? So someone has to replace it. Who will replace? Water molecule will replace. You are getting the point. So this mechanism called as what? Photolysis of water. Photo light. In the presence of light, lysis, breaking down of water. This is called water splitting complex or oxygen evolving complex. When one water molecule break down, there is a release of two proton, two electron, and how many? Half. So they will directly ask how many number of oxygen molecules are released when 10 protons are break down. How many will release? 5. Only half oxygen will be released. Now again, the question which is asked multiple times, which of the following ion can act like a cofactor for the, not magnesium, which is the manganese and chlorine, sometimes even calcium. Okay, sometimes. Okay, but majorly is manganese and chlorine. Correct? Now don't confuse between magnesium and manganese. Now do not get confused with the point. Okay, this is only more important among all. Oh, so much fast. Okay, now the question is here, where the photolysis of water is taking place? That is the question. See, can you see friends, this is stroma, thylakoid membrane, this is lumen. What's happening here? Water molecule breakdown, there is a release of proton. Protons released into lumen. What about the oxygen? This is what we are breathing. This we use it for cellular respiration. So no plant is releasing the oxygen purposely. To fulfill their deficient electron, they are just breaking down the water. That's why they are releasing oxygen. Now what is our goal here? Electron. Correct na? Who will take the electron now? P680 will take the electron. No point of confusing over there. Now another important point here, in case of cyclic photophosphorylation, we produce only one ATP, that's all. But in non-cyclic, definitely we produce NADPH also, ATP also. But in this picture, I have shown you only the production of NADPH. But compulsory we produce ATP also. The production of ATP mediated by PMF. What is that? MF. Proton motive force. By using proton motive force, we can synthesize the ATP. How ATP can be synthesized in the non-cyclic photophosphorylation explained by an important, what we can say that, pathway named as chemiosmotic pathway. Okay, what is the name of the pathway? Chemiosmotic pathway. So, chemiosmotic hypothesis has been put forward by Peter Mitchell, which is also called as a Mitchell hypothesis. Okay, he is the one who explained ATP synthesis can be exclusively mediated by the proton motive force. Correct na? Now, all of you just look into this picture and try to understand. We will prove this very clearly. If ATP want to be synthesized, this is the direct NCERT line and we get the question. If the ATP want to be synthesized, we require proton pump, also called as, okay, we'll see proton pump, then proton gradient, ATP synthase are the three major requirements for the ATP to synthesize. What are those? Proton pump, proton gradient. What is called gradient? Moving from high concentration to the low concentration, and ATP synthase. Now first let me prove, later we can read this easily, you can answer without looking also. Okay, I'll explain slowly, focus on the picture so clearly without having a confusion. This is all about understanding. This is thylakoid membrane. Can you see P680, P700, ATP synthase enzyme, all electron acceptor complexes. Correct na? Now friends, focus and keep on answering to me. First, P680 absorb the energy, release the excited electron. Correct na? Who will accept that? First one. Since again you are talking about cyclic, we are talking about what? Non-cyclic. What is that? Don't forget, pheophytin. First class now you people get sleep. Look into this. What is that? Pheophytin. Correct or not? After pheophytin? Okay, no problem. No problem, no problem. Look into this. In case of a cyclic, who is the first electron acceptor? Ferredoxin, plastoquinone, cytochrome B6F and plastocyanine. Correct na? In case of a non-cyclic, first P680, pheophytin, plastoquinone A and B, cytochrome B6F and plastocyanine. Clear this one? Now tell me, first from P680, electron got excited. Who accepted it? Who accepted it? What happened here? Okay, pheophytin, very, very good. Then pheophytin transfer the electron to where, friends? Very good. Then look into the point, don't get confused. Upon accepting the electron from the pheophytin, plastoquinone reduced. What? Plastoquinone reduced to PQH2. What happened? Okay, it is reduced to what, friends? PQH2. Now interestingly, if the plastoquinone want to reduce into PQH2, along with the electron, it also require proton. You are getting the point? Along with the electron, it is also required proton. So from where the protons are coming from? Stroma. Stroma is full of protons. Now first once again tell me, first P680 excited the electron, accepted by? Then from pheophytin who will accept? Then plastoquinone accept electron from pheophytin, proton from? What happened to this pen all of a sudden? Okay, accept proton from the stroma, then plastoquinone reduce it to PQH2. But plastoquinone, PQH2 never keep the electron with it. Correct na? It will transfer to what next? Cytochrome B6F. Correct na? Then can I say PQH2 oxidized back to PQ? Correct na? So, but where is the proton? Don't get, no, only electron transferred to the cytochrome B6F. Proton released into the lumen. You got the point? What happened to this all of a sudden? Nothing like that, ma. Okay, what happened? Proton released into what? What is that first pen? This one now. Okay, proton released into where? Lumen. Now once again quickly understand, first pheophytin send the electron to plastoquinone. Plastoquinone upon accepting electron from pheophytin and proton from stroma reduced to PQH2. Then upon releasing electron to cytochrome B6F, PQH2 oxidized back to PQ. Correct na? Then what about that proton? Released into lumen. So these processes keep on happening. Then what happens? Protons from the stroma, where they are entering into? Lumen. Just now we discussed when water molecule break down, what happened? We got electron, we got oxygen and proton. So can I say the concentration of proton become extremely higher in the lumen compared to the stroma? So to maintain the charge balance, all the protons are moving from the lumen to the stroma. Okay, whenever protons are moving, they are always move in the form of a wave like this. Always remember, electron lose energy, proton generates energy. When proton moved where to where? Stroma, lumen to stroma. Now proton came out. Then there is a generation of energy. That energy can be used to join ADP with PN. Then there is a production of ATP. Correct na? Now tell me, this is which direction? Stroma. Can I give you a statement? Production of NADPH and ATP occurs towards the stromal side.
Yes, sir.
Correct na? And protons always move from lumen to stroma. First they move from stroma to lumen, then they move from lumen to stroma. So in the non-cyclic we produced pH and ATP. Correct na? Now tell me friends, in the production of ATP, which photosystem is involved? PS2. Correct or not? In the production of NADPH? PS1. Since both photosystems are present, we produce both. In bacteria, we have only one photosystem, so we produce only ATP. Correct na? No point of getting confused over there. Okay. Okay. Answer this. This is 2020 paper. Slowly. Plastoquinone. So when you get this question, write it. In the exam, you recall it.
Cytochrome B6F. Option B.
Option? What, which option? Right or wrong? There is no point of making wrong. Look into that. Plastoquinone. Write it, friends. Facilitate the transfer of electron. So they have not said whether it is a cyclic or non-cyclic. So first we don't know, no. Let's write it. First ferridoxin, then plastoquinone, then cytochrome B6F, then plastocyanin. Correct na? This is where, friends? Cyclic. Correct or not? Let's write non-cyclic. In the non-cyclic, very good, PS2, pheophytin, then plastoquinone, same na, and plastocyanin. Now what is the question they are asking? Plastoquinone facilitate the transfer of electrons from. When I use the word from, electrons are generally coming from where? PS2. And electrons are moving. See, where is the PQ is located? Here. Correct na? After the PQ, what is there? So it is moving electrons from, you can say like this, pheophytin to cytochrome, or you can also say PS2 to cytochrome. But they have not given pheophytin in the option. They have given? No. Then where they have given pheophytin? Where they have given pheophytin, I am saying. Pheophytin they have not given na. Then what they have given? Correct, ma, see here, plastoquinone is located between what to what?
Pheophytin to cytochrome.
So, but they have not given the option of pheophytin. Then obviously electron is coming from where? PS2 to PQ. PQ to cytochrome. So, PQ is located between PS2 and cytochrome B6F. Correct or not? Correct, na? Don't get confused. Slowly you have to think and answer the questions. What is this? highest number or protons are found in? We no need to think, right? Lumen only, na? From stroma it is releasing into lumen. At the same time water molecule is also breaking down in the lumen only. So obviously lumen contain highest number of thylakoids. Correct, no? Correct, no? So this is light reactions. Now I want you to take out this PPT and read very elaboratively, okay, till light reactions. So tomorrow we'll continue the dark reactions, okay. I'll teach very slowly. By October or November I'll complete the entire syllabus of biology, okay. So other madam also teach botany and zoology. As a result, whole subject will revise twice. So you keep the target of 340 and above that, okay. By that we can crack it easily, okay. Now the only thing I am telling you, I don't tell many number of times. See, the only difference between successful candidate in the NEET and non-successful candidate in the NEET is only simple qualities. The number one is consistency, okay. And if you want to be consistent, you have to be disciplined much. If you want to be disciplined, first you have to get interest. Okay, with interest you automatically get disciplined, because if you like the work what you do, automatically you get disciplined, and with consistency you can crack the exam. To crack the NEET you no need to be intelligent. There is a simple formula: 98% you have to do hard work and 2% you require intelligence. Undoubtedly you can crack the exam, okay. All right, okay, we'll continue in the next class, okay. In the chat option of your app I will upload the PDF, okay. In the app you can see the chat option, no? If you click on the chat option you can download automatically, okay. Mandately revise it, then we will do it, okay. You tell me, we'll do 2 units simultaneously or first we'll complete 1 unit? I am thinking we will start human physiology. If you are okay with single unit, we will go ahead, or 2 units simultaneously also we can do. We'll complete single unit. Perfect. All right. Okay. All right. Done. Okay.